So I'm gonna start by introducing myself. So my name is Eduarda. I'm a co-host at the BordeauxTea and the Amsterdam Tea, and I do my PhD at the University of Bordeaux. And I also work as a research assistant at the VUMC in Amsterdam. Dana Collins is the BordeauxTea co-host. She also does her PhD here in Bordeaux. She researches the neurobiology of addiction. Emil is the VU Amsterdam Tea co-host, so he also studies and does his PhD at the VU, and he is researching methods to improve the statistical power in GWAS studies. And finally, Dana, our guest today, she is an instructor in human and molecular genetics at the Virginia Commonwealth University, and she is also the reproducibility co-host of the local university. So first, I would like to ask everyone here in the chat, in the meeting, to put in the chat where you are, or where you work from, and which field of research or in what are you working. The idea is that this meeting should also be used to give the possibility that people can network between different universities and different programs. So really feel free to use this meeting to sort of reach people that might have same interests as you. And this meeting will be recorded only for the presentation part, and will go to YouTube, but the discussion won't be recorded and it won't go to YouTube, so that you have more freedom to talk and discuss if that could bother you in any way. Now, I give the floor to Dana, and thank you again for accepting this exchange experience with us.
Thank you so much. The pleasure is all mine. I'm super excited to do this exchange. So today I'm going to talk about flexible analysis, and I'm going to do it by focusing on a paper called Many Analysts, One Dataset. A little bit about me. So I am a classically trained geneticist. Before I came to academia, I was actually working in a clinical laboratory. And then when I came to the university, I sort of have two main roles. In my academic role, I am an instructor in a data science course, and I'm also I have the privilege to co-host the reproducibility group at VCU. One of the other co-hosts is here, Nina Exner. So good to see you. And then research-wise, I have focused on DNA methylation, and particularly looking at patterns related to birth outcomes, like preterm birth and major depression. The reason I bring up the research aspect here is because it ties really well into our topic today. And if you're not familiar with DNA methylation research, don't worry at all. All you have to know is that it is a hot mess. So what do I mean by that? Well, there are basically no standards in DNA methylation literature as far as what is the best practice for data cleaning. Which steps should you do, and what order should you do them in? No hard and fast rules about covariate selection or even multiple testing correction thresholds. Some people use Bonferroni, some people use FDR, some people don't use any. And this can be very frustrating because in the literature, when you see two results that seem to be in conflict, or at the very least they're inconsistent, it can be difficult to determine if that inconsistency is due to methods differences in the statistical handling, or if it's something more fundamental. Maybe there was a difference in study design, like demographics or study bias, or maybe the first report was a false positive. It's difficult to tell. And that's what got me thinking about, well, how do we quantify the impact of hundreds of choices, big and small, on individual analyses, and on the robustness of those analyses and the credibility of the literature as a whole? And by no means am I the first person to talk about this. Definitely want to give a nod to Andrew Gelman. He's got a lot of really good papers out there. And he talks quite a bit about the garden of forking paths, these data-dependent analysis decisions. because they can increase the likelihood of false positives. And there are, as I said, many, many papers out there. When I discuss these sorts of topics with the upper level molecular HGen students, a lot of the genetic students want to focus on the chemistry aspect. You know, maybe it was a reagent difference, maybe it was biological replicates or technical replicates. And there's just sort of this hope and prayer that all of the differences, you know, it comes down to the mice are acting differently, or maybe there was something to do with variability in data collection. And really, if we all could start from the same data, we'd all end up in the same place. That's the hope, the dream. And that's why I really like this paper, because it starts us off with everyone having the same data. And it's especially nice because we can focus just on the statistical handling from having the data. There's no jargon, you know, we're talking about football or soccer. And as far as, you know, some of the other papers, like if we did a DNA methylation paper, we'd have to all be familiar with DNA methylation methods. And so focusing on something like this data set really helps us hold interdisciplinary discussions. So here was the setup. There were 29 teams, and they all got the same prompt. And the prompt was, do soccer referees give more red cards to dark-skinned players than light-skinned ones? And they all had the same data. And here is a figure sort of detailing the main result from the paper. So what you're looking at here, each circle is a point estimate from an individual team's statistical model. The rectangles, these gray rectangles, that's the 95% confidence interval. And then if these circles are green, it was statistically significant for that individual team's model. And if it's gray, it wasn't statistically significant. And so what we can see is that about 2/3 of the team report that they found statistically significant results, and that pretty much all of the results are significant in the same direction of effect, towards referees being more likely to give red cards to dark-skinned players. But we can see that the magnitude of the point estimate and the certainty around the point estimate varies quite a bit from team to team. And if we dig deeper and try to figure out why this happened, the first place I always look is covariates. Sometimes these are cheekily referred to as researcher degrees of freedom. Sort of keep adding them in until you get what you like. And what we can see here is that the number running along here, this is the number of covariates chosen by each team. It varies from 0 covariates to 7. And then there isn't a single covariate that every team chose. And even, you know, player position is the most commonly, but after that, it really becomes far more sparse. So very little consistency in covariate selection. We look at analytic approach. On the right, we see the same figure as before, but now instead of ordering it by point estimate, we are ordering it by the general kind of model used. And we see here that there is a wide variety, again, of model choices, ranging from a sort of complex model where you have a multi-level logistic regression with Bayesian inference, to they use Spearman correlation, or maybe a Poisson regression or a zero inflated Poisson. Lots and lots of variability. And so what does this level of variability mean for research? Well, I hope we talk about that quite a bit in a minute. But I would argue that at minimum, this really reinforces that transparency is critical. Even if you are working on data that you are not allowed to share, sharing your scripts is huge and should be expected. Pre-registration, so the act of writing down what you're going to do before you have any data, that may help save us from ourselves. It's a thing that in the US, all clinical trials are required to do. to again, try to encourage people to declare ahead of time and make transparent what they are expecting. And then methods like the multiverse analysis. I think this is probably where we're going to start moving. So these methods help us accurately, or at least more accurately, categorize the robustness of a result. And how it works is we take that garden of forking paths, and rather than reporting just one possible analysis strategy, or even planning ahead, oh, I'm only going to do this one, you say, okay, these are all of the defensible analysis choices that a reasonable person with this research question and this data set would make. I will do all of the permutations of what is possible, and I will report all of the outcomes so that my readers can see exactly how robust my findings are. Is it sensitive to a certain covariate or a certain outlier? That's I think where we'll probably start going. But again, I'd love to hear everyone else's thoughts. And then, just to get some people thinking, I threw a few questions up here. But that is all I have for my presentation. And I am excited to discuss this further.